Hi guys, today's topic is chapter number 2, sexual reproduction in flowering plants. Contents are the parts of the flower, pre-fertilization, double fertilization, the post-fertilization, apomixis and polyembryony. The first topic is parts of the flower. To a biologist, flowers are morphological and embryological marvels and the site of sexual reproduction. The parts of flower is divided into essential organs and non-essential organs. Essential organs include androecium, that is stamen, and gynoecium, that is carpels. Non-essential organs include calyx, that is sepals, corolla, that is petals. Let's learn about the pre-fertilization structures and events. The first one is stamen, microsporangium and pollen grain. Stamen has two parts, a long slender stalk called filament and the terminal bilobed structure called anther. The proximal end of a filament is attached to a thalamus or the petal of a flower. The number and the length of the stamen vary from one species to other. A typical angiosperm anther is bilobed with each lobe having two theca, that is, they are dithecous. A longitudinal groove runs lengthwise separating the theca. The anther is four-sided, that is tetragonal structure consisting of four microsporangia located at the corners, two in each lobe. Microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Let's learn the structure of microsporangium. In a transverse section, typical microsporangium appears circular in outline. It is generally surrounded by four wall layers: the epidermis, endothecium, middle layer and tapetum. The outer three wall layers perform the function of protection and helps in dehiscence of anther to release the pollen. The term dehiscence means splitting up. The innermost wall layer is called as tapetum. It nourishes the developing pollen grains. The cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. Let's learn about microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergoes meiotic divisions to form microspore tetrads. What are microspore tetrads? Microspores arranged in a group of four. As each cell of a sporogenous tissue is capable of giving rise to microspore tetrad. Each microspore tetrad is a potential pollen or microspore mother cell, shortly called MMC. The formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. As the anther mature and dehydrate, the microspores dissociate and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with dehiscence of anther, that is splitting of anther. Let's learn about pollen grains. The pollen grains represent the male gametophyte. Pollen grains vary in sizes, shapes, colors, designs from different species. They are spherical, measuring around 25 to 50 micrometers in diameter. It has prominent two-layered wall, the hard outer layer called as exine, which is made up of sporopollenin, which is one of the most resistant organic material known. It has withstand high temperature, strong acids and alkali. Pollen grain exine has prominent apertures called germ pores, where sporopollenin is absent. Pollen grain also welds fossils because of sporopollenin present in it. The inner wall of pollen grain is intine, which is thin and continuous layer made up of cellulose and pectin. The inner wall of the pollen grain is surrounded by a plasma membrane. When the pollen grain is matured, it consists of two cells: vegetative cell, generative cell. Vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. Generative cell is small and floats in cytoplasm. In over 60% of angiosperms, pollen grains are shed at the two-celled stage. The 40% of remaining species the generative cells divides mitotically to give rise to two male gametes before pollen grains are shed, that is 3-cell stage. Pollen grains of many species causes severe allergies and bronchial affection. Example, asthma and bronchitis. Parthenium, otherwise called as carrot grass, that came to India as a contaminant with imported wheat is main cause of pollen allergy. Pollen grains are used because it is rich in nutrients. It is used as pollen tablets for food supplements. In Western world, pollen production is used in form of tablets and syrups which are available in market. Pollen consumption increases the performance of athletes and race horses. When once they shed, pollen grains land on the stigma before they lose viability, which bring about fertilization. The period for which the pollen grain remain viable is highly variable and to some extent depends on temperature and humidity. For example, species like rice and wheat, the viability is around 30 minutes of their release. Example of other species like Rosaceae, Leguminosae and Solanaceae, the viability is around months. Pollen grains of large number of species can be stored in liquid nitrogen, that is minus 196 degree. Such stored pollen can be used as pollen banks similar to seed banks in crop breeding programs. It is also called as cryopreservation. Let's talk about another topic, the pistil, Megasporangium, that is ovule and embryo sac. Gynoecium represents the female reproductive part of the flower. It consists of single pistil called monocarpellary or may have more than one pistil called multicarpellary. When there are more than one pistil fused together, it is called as syncarpus, or if it is free, it is called as apocarpus. Each pistil has three parts: stigma, style, ovary. Stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulged part of a pistil is the ovary. Inside the ovary is the ovarian cavity, which is nothing but locule. The placenta is located inside the ovarian cavity. Arising from the placenta are the megasporangia, commonly called ovules. If it is one ovule, the examples are wheat, paddy and mango. If there are multiple ovules, the examples are papaya, watermelon and orchids. Now let's learn about megasporangium, which is nothing but ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in a region called as hilum. That is, hilum represents the junction between the ovule and the funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the ovule except at the tip where a small opening called the micropyle is organized. Opposite the micropylar end is the chelaza, which is the basal part of the ovule. Inside the integuments is the mass of cells called as nucellus, which have reserved food materials. Inside the nucellus is the embryo sac, or this otherwise called as female gametocyte. An ovule has a single embryo sac formed from megaspore through reduction division, that is meiosis. Let's discuss about megasporogenesis. The process of the formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules differentiate a single megaspore mother cell in a micropylar region of the nucellus. The MMC undergoes meiotic cell division, which results in production of 4 megaspores. In a majority of flowering plants, one of the megaspores is functional whereas other degenerate. The one which functional megaspore develops into a female gametocyte. This method of embryo sac formation from a single megaspore is termed as megasporic development. The nucleus of a functional megaspore divides mitotically to form two nuclei which move to the opposite poles, forming two nucleate embryo sac. Two more sequential mitotic nuclear divisions result in the formation of 4 nucleate and later the 8 nucleate stages of the embryo sac. After 8 nucleate stage, The cell walls are laid down, leading to the organization of typical female gametocyte or embryo sac. 8 nuclei splits into 6 nuclei and 2 nuclei. The six nuclei are surrounded by the cell walls and organized into cells, whereas the 2 nuclei are the polar nuclei situated below the egg apparatus in the large central cell. The 3 cells are grouped together at the micropylar end and constitute the egg apparatus. Egg apparatus consists of 2 synergids and 1 egg cell. The synergids have special cellular thickenings at micropylar end called filiform apparatus, which guides the pollen tubes into the synergids. 3 cells at the chalacal end are called as antipodals. The large central cell has 2 polar nuclei, whereas a typical angiosperm embryo sac at maturity, which has 8 nuclei but it is 7-celled. Let's learn about pollination. Transfer of pollen grain to stigma of a pistil is called as pollination. There are 3 kinds of pollination: autogamy, geitonogamy, and xenogamy. Autogamy is otherwise called as self-pollination. Pollination that is achieved with the same flower. In normal flower, which opens and exposes the anthers and stigma, complete autogamy is very rare. Autogamy in such flowers requires synchrony and stigma receptivity, and the fact that anther and stigma should lie close to each other. For example, the plants like oxalis, camelina produce 2 types of flowers. The plasmogamous, which with exposed anther and stigma, whereas cleistogamous, which doesn't open at all. So, cleistogamous flowers strictly undergo autogamy. Now let's learn about geitonogamy. Transfer of pollen grains from the anther to the stigma of another flower of a same plant is called as geitonogamy. It is genetically similar to autogamy, but functionally it is cross-pollination. Now let's learn about xenogamy. Transfer of pollen grains from the anther to the stigma of another or the different plant is called as xenogamy. It is only type of pollination which genetically brings different types of pollen grains to stigma. Now let's learn about agents of pollination. There are 2 agents of pollination: abiotic and biotic. Abiotic examples are wind and water. Biotic examples are animals and insects. Only a small portion of plants use abiotic, whereas majority of plants use biotic factors. To compensate the uncertainties and the loss of pollen grain, the flower produces enormous amounts of pollen grain compared to the number of ovules available for pollination. Now let's learn about pollination by wind. It is the most common abiotic pollination. It requires that the pollen grain should be light and non-sticky. They often possess well-exposed stamen, which is easily dispersed in wind, and large feathery stigma to trap pollen grains. It is very common in grasses. Now let's learn about pollination by water. Quite rare in flowering plants, which is limited to 30 genera, mostly monocots. What is the regular mode of transport among male gametes? Among lower plants like algae, bryophytes and pteridophytes. Examples are Vallisneria, hydrilla, sea grasses called zostera. Not all aquatic plants pollinate through water. The water hyacinth and water lily, the flower emerge above the water level and is pollinated by insects or birds. In most of the water pollinating species, pollen grains are protected from wetting by a mucilaginous covering. Majority of flower plants use a range of animals as pollinating agents. Examples: bee, butterflies, flies, beetles, wasps, ants, moths, birds such as sunbirds and hummingbirds, and bats. Bees are dominant biotic pollinating agents. Even larger animals like lemurs, rodents, reptiles like gecko and garden lizard are also pollinators in some species. Flowers of animal pollinated plants are specifically adapted for a particular species of animal. Majority of insect pollinating flowers are large, colorful, fragrant and rich in nectar. Animals are attracted to flower by a color or fragrance. The flowers pollinated by flies and beetles secrete a foul odor to attract these animals. To sustain animal visits, flowers have to provide rewards to animals. Nectar and pollen grains are usual rewards. Visiting animals comes in contact with the anthers and the stigma, which further helps in pollination. In some species, floral rewards are providing safe space to lay eggs. For example, Amorphophallus, a tallest flower, which is 6 feet. Similar relationships are seen in the species of moth and plant Yusa. Now let's discuss about outbreeding devices. Many flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of same flower. The continued self-pollination result in inbreeding depression. Flowering plants develops many devices to discourage self-pollination and encourage cross-pollination. In some species, pollen release and the stigma receptivity is not synchronized. Either the pollen is released before stigma becomes receptive, or stigma becomes receptive much more than the pollen could release. In some species, anther and stigma is placed in different positions so that the pollen cannot come in contact with the stigma of the same plant. Both these devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. This is genetic mechanism and prevents self-pollen from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unicellular flowers. In male and female flowers present in the same plant, such as castor and maize, which is monoecious, it prevents autogamy but not geitonogamy. In papaya, which is dioecious, it prevents both autogamy and geitonogamy. Let's discuss about pollen-pistil interaction. All events from the deposition of pollen and stigma till the pollen tube enters the ovules called as pollen-pistil interaction. Events includes the recognition of compatible pollen, germination of pollen grains, development of male gametophyte. The compatible events or the right type includes pistil accepting pollen, formation of pollen tube reaches ovary, pollen grains moves into pollen tube enters ovule through micropyle, pollen tube enters synergids through filiform apparatus, the fertilization occurs. Whereas incompatible events or the wrong types include pistil rejecting pollens, which prevents pollen tube growth and prevents fertilization. This interaction is mediated by chemical components of pollen and pistil. The pollen-pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of pollen. Now let's discuss about artificial hybridization. The major approach of this process is for crop improvement program. In such crossing experiments, it is important that only desired pollen grains are used for pollination and stigma is protected from contamination, that is unwanted pollen. It is protected by two techniques, emasculation and bagging. Removal of anthers from flower bud before an anther deceases in a bisexual flower bearing a female parent using a forceps is called as emasculation. Emasculated flowers have to be covered with a bag of suitable size made up of butter paper to prevent contamination of its stigma with unwanted pollen. This process is called as bagging. When the stigma of bagged flower becomes receptive, the collected pollen grain are dusted onto the stigma and then the flower is rebagged. Here fruits are allowed to develop. In a female plant, which is unisexual, emasculation is not necessary. Here, female bud is directly bagged and when the stigma turns receptive, suitable pollen grains are dusted onto it so as to allow germination. Now, let's discuss about double fertilization. When the pollen grains fall on the stigma, the pollen tube enters one of the synergids and releases two male gametes. One of the male gametes moves towards the egg cell, fuses with it to complete the syngamy to form the zygote. The other male gamete fuses with the two polar nuclei and forms triploid, that is primary endosperm nucleus. This is termed as triple fusion. Since two kinds of fusion, syngamy and triple fusion, takes place, the process is known as double fertilization, as it is characteristic of flowering plants. After triple fusion, the central cell becomes a primary endosperm cell. The primary endosperm nucleus gives rise to the endosperm, while the zygote develops into the embryo. Now, let's learn about the post-fertilization events. It includes the development of endosperm and embryo and maturation of ovules into seeds and ovaries into fruits. Let's discuss about the formation of endosperm. The endosperm develops before the embryo because the cells of the endosperm provide nutrition to the developing embryo. The primary endosperm nucleus repeatedly divides to give rise to free nuclei. This stage is called as free nuclear endosperm. Cell wall formation occurs next, resulting in a cellular endosperm. The endosperm may be either fully consumed by the growing embryo, that is peas and beans, or the portion of the endosperm is retained in the matured seed. Example is coconut and castor. Let's discuss about the development of embryo. The embryo develops at the micropylar end of the embryo sac, where the zygote is situated. The zygote gives rise first to the proembryo and then to the globular, heart-shaped mature embryo. A typical dicot embryo consists of embryonal axis and two cotyledons. The portion of the embryonal axis above the level of the cotyledons is called as epicotyle. Epi means above. It contains the plumule. The portion below the axis is called as hypocotyle. Hypo means below. The root tip is covered by the root cap. In a monocot embryo, there is only one cotyledon. In grass, it is known as scutellum and is situated at one side of the embryonal axis. At its lower end, the embryonal axis has the radicle and the root cap enclosed in the coryza. What is coryza? It's undifferentiated sheath covering radicle and root cap. The epicotyle lies above the level of the scutellum and has the shoot apex and the leaf primordia enclosed in hollow structures called as coleoptiles. Coleoptiles is nothing but the sheet covering the plumule. Now let's discuss about the development of seeds. It is the last stage of sexual reproduction in angiosperms. Seeds are of fertilized ovules that are developed inside a fruit. A seed consists of a seed coat, cotyledons, embryonal axis. Seed coat has two layers, a thick outer layer called as testa and a thin inner layer called as tegmen. Seeds may be albuminous, where endosperm is present. Examples are wheat and maize, or non-albuminous, where endosperm is absent. Examples could be groundnuts. Some seeds such as black pepper and wheat have remnants of nucellus known as perisperm. The integuments of ovules harden to form seed coat, and the micropyle facilitates the entry of oxygen and water into the seed. As it loses moisture, the seed may enter dormancy, or if the favorable conditions exist, it germinates. Now, the last part is the development of the fruits. The ovary of the flower develops into a fruit. The walls of the ovary transform into the walls of the fruit. Example is pericarp. Fruits may be fleshy, as in mango and orange, or can be dry, as in groundnut and mustard. In some plants, floral parts other than the ovary take part in fruit formation, as in apple and strawberry. In these, the thalamus contributes to fruit formation. Such fruits are called as false fruits. Fruits that develop from the ovary are called as true fruits. Examples are tomato, grapes, and banana. Some plants develop without fertilization and are known as parthenocarpic fruits. Examples are banana. Let's learn about the last topic called as apomixis and polyembryony. Some plants produce seeds without fertilization. This process of seed formation is called as apomixis. Apomixis is a form of asexual reproduction mimicking the sexual reproduction. In some species, apomixis occurs as a diploid cell informed without meiosis and develops into embryo without fertilization. In some varieties like citrus fruits and mango, the nucleus cells divide and protrude into an embryo sac to develop into embryos. In such cases, each ovule may contain several embryos, and this condition is called as polyembryony. Apomixis is important for producing hybrid varieties of fruits and vegetables and also for increasing crop yield multifold. Thank you.